Hi, my name is Sam Thibodeau, and my research project focuses on the inhibitory effects of curcumin on human embryonic kidney HEK 293 cell growth. So what exactly am I going to be researching? So the focus of this study is to determine the inhibitory effects that curcumin has on cell cancer growth, specifically through a proliferation assay and a wound healing assay, using varying concentrations of curcumin. I'll also be testing the activation levels of the P53 and NFKB pathways, which are known to regulate cell growth, and in cancer they're downregulated. So I'm testing to see if curcumin upregulates them. So I will be doing a qPCR test to determine the activation levels. And so the reason I'm doing this is because in past studies, curcumin has been shown to inhibit growth of cancer cells in both a proliferation and a wound healing assay. And so my study will aim to build on data by exposing the HEK cells to different and higher levels of curcumin. And because of my past research, my current hypothesis is that curcumin will increase P53 and NFKB levels, which will allow for a greater regulation of cancer cell growth in both the proliferation and wound healing assay. So just some background research on what curcumin is and HEK cells. Curcumin is actually the active polyphenol in the spice turmeric, which turmeric is both a spice and a plant known as Curcuma longa. And turmeric has been used in medicine for thousands of years, specifically in Southeast Asia. It has a lot of great properties. It's antioxidant, anti-inflammatory, antibacterial. But they didn't know exactly where those properties came from until more recent studies have actually attributed those properties to curcumin, and curcumin has demonstrated antimicrobial, anti-inflammatory, anti-cancer properties, and it actually targets multiple cell signaling pathways. And so HEK cells, HEK 293, they're super commonly used strain in biologies. Mostly because they have a super fast doubling time and they're easy to maintain, and they're used in a lot of cancer studies because they have they're still tumorigenic like a lot of cancer cells, easy to maintain, which some cancer cells aren't necessarily, and they have a really fast doubling time, about 34 to 36 hours, so they can get studies done really fast. And so curcumin has been shown to reduce cancer growth and induce apoptosis with human breast cancer, human pancreatic cancer, kidney cancer, both in vitro and in vivo. And it's been shown to upregulate and downregulate a variety of signaling pathways that are essential to the cancer growth, including the NFKB and P53 pathways and P65. So currently I am in the process of my two different assays, the cell proliferation assay and the wound healing assay. For my cell proliferation assay, I am doing an MTT colorimetric assay, which, dependent on cell growth, when the cells are exposed to an MTT solution, they will change to a shade of purple, which I can then read in a microplate reader at 545 nanometers, which will tell me how well they grew. So to do so, I will be seeding the cells at 2,000 cells per well in a 96 well plate. The cells will be in complete media consisting of DMEM, 10% FBS, 1% strep pen, and then varying levels of curcumin, 0, 10 micromolars, and 50 micromolars. And then certain cells at different days will get exposed to the MTT solution. So some will be at 2, some will be at 4, some will be at 6. And at those, the MTT solution will be added, incubated for 2 hours in that same 37 Celsius incubator with 5% CO2. And then after those 2 hours, the cell suspension will be measured at 545 nanometers in a microplate reader, and the absorbance of that will reflect the cell growth and tell me how much the curcumin inhibited the growth. For my wound healing assay, I have a 6 well plate where the HEK cells will be seeded at 40,000 cells per well. They'll be cultured in the same DMEM with 10% FBS and 1% strep pen, along with that same 0, 10, and 50 micromolars of curcumin. And so the cells will grow originally just in that DMEM, 10% FBS, and strep pen, and then at 70-80% confluence, a slit is made through the cells, and then they're exposed to that 0, 10, and 50 micromolar concentrations of curcumin, and that's for 48 hours. and at 0, 24, 36, and 48 hours, I would be looking at those cells in a microscope and kind of just determining to see the inhibitory effects, taking photos of those and kind of comparing them to see how well the slit closed up when in the presence of different concentrations of curcumin. So just overall, this study will build on the current research out there with curcumin in regards to cancer. It'll see, should we continue to test curcumin against different cancer strains in combination with other drugs to see if curcumin can still help with, the tumors and reducing the size of the tumors. For me, my future plans are, again, determining the activation levels of the NFKB, P65, and P53 levels using qPCR. I also want to repeat the same wound healing and cell proliferation assay, but this time with a positive control that's known to inhibit cancer growth to see how effective the curcumin was or wasn't, again, as a method to inhibit the growth. And then also, testing different cell lines to see, the HEK cell line will show one thing, how will other cell lines that are cancerous compare? And all of those will just kind of continue to build on the research we have. I can continue to make conclusions and continue to see whether curcumin can be a valuable asset in cancer treatment in the future.